Okay, congratulations. You have made it this far. You've completed the color wheel. You have completed extracting your own DNA. And now let's look at this piece. How do you analyze DNA in order to identify people? Well, the first piece you've done. You've got to extract it. So think about how you did that, right? You happened to take a cheek cell. This picture shows a hair. But basically, you need to use chemicals in order to remove the DNA and to isolate it from the other fluids and parts that make up the body of the cell. The next piece is amplification. So think about what that word means, amplification. You need to take part of that DNA and you need to amplify it. So you need to make it much bigger in order to see it. Because remember, a lot of our DNA code is identical to each other. And in fact, we share huge amounts of our same genetic coding with a banana, believe it or not. So a lot of this stuff is more similar than you would think. But we want to look at the codes that make us unique as a species. And then within this, we want to look at the code that makes us unique from one individual to the next. So that's amplification. So you're taking a part of that DNA and you're making a copy of that piece over and over and over and over again. And we're about to watch a video that will guide you a little more into that so that we can actually see that. And then your third piece is visualization. So how can you visualize this to see how it matches from one to the next? And the way you're going to do it is by running a gel. So DNA is separately loaded into each one of these little wells here. And then it runs through the gel. We'll talk about it more later. And that's going to give you a sense of how do these patterns match up. Okay. So for our next slide here, DNA extraction. Again, you've done that. So you just extracted your own DNA and got to look at it. Now for amplification, okay? So now I'm going to run the video to give you a little better sense of what it means when you are running this machine called a mini PCR.
PCR is a powerful technique that allows us to make billions of copies of specific pieces of DNA. The first step in PCR is to open up the two strands of DNA to expose the genetic information encoded by the nitrogen bases, A, C, T, and G. In the next step, custom-made DNA molecules help us find the beginning and end of the DNA piece of interest among the billions of letters in the genome. These genetic bookmarks are called primers. The third and final step is to fill in the genetic information between the primers.
So just to notice here that you're actually, some of the chemicals in your tubes that you're going to receive, one of them is a primer. So you're going to actually put primer into your DNA to cause it to start to find that.
Enzyme called DNA polymerase adds the building blocks of DNA called nucleotides.
Okay, so then once the primer comes back in, then you've got to actually have this filled in, and that's when the DNA polymerase comes around and does that.
To create an identical DNA strand, DNA polymerase works in our own cells to replicate our own DNA each time a cell divides. The first step in PCR is known as denaturation and is enabled by high heat. The second step is called annealing, and different primers have different and very specific annealing temperatures, typically between 50 and 60 degrees Celsius. The third step is called extension. DNA polymerase used in PCR works best at 72 degrees Celsius.
So again, first you get this piece of DNA in a tiny little segment. It gets opened up, right? And then the primers come in and match exactly what it is on the other side. So where you had one strand, now you have two, and then you actually fill in the rest of the copy with this DNA polymerase. So that finishes it up. So now we had this one strand of DNA, one section, and now we have two.
It's best at 72 degrees Celsius. In these 3 steps, we've taken our original DNA molecule and obtained an identical copy of a very specific fragment.
So as you run this cycle in your machine, every time the cycle goes up and down, you make a copy. So let's look at how those copies are going to multiply.
If we repeat these 3 steps one more time, we'll go from 2 copies to 4. Repeat it again and we'll obtain 8 copies. And again, and we'll get 16, and 32, and 64, 128. Well, you get the idea. DNA amplification by PCR is an exponential process. After 30 copy cycles, we will have 2 to the 30, or 1 billion identical DNA copies.
So that is some serious amplification, where you go from 1 to a billion of that segment of your DNA, and that's what you're going to do in the Missy Baker lab.
This is enough DNA, for example, to detect a pathogen in blood such as HIV or Ebola, or to amplify trace amounts of DNA found at a crime scene, or to enable any of the other powerful applications.
Okay, so now you've seen the video. So we've got DNA amplification down, I hope. You have a sense of it, where you take one segment of the DNA and you make billions of copies of just that one unique segment. Now we'll go on to DNA visualization. So now we have it amplified. What are we going to do with it next? So that's where you're going to actually take it and you're going to run it through the gel. And to do that, you're going to actually be doing a lab where you're going to see how DNA moves through the gel, just like ink will move through paper. Sounds a little mysterious, but we'll be doing that after you run your PCR. Okay. All right, there you have it. Now you're going to be doing it CSI style. It's time to start the micropipetting, running the PCR. There's your PCR machine. Then you're going to make it go through gel electrophoresis. You put it through the gel. Our gel machines look a little different than that. Then you will analyze the gel and see how that DNA matches up. All right.